Today, we will discuss about smooth muscle physiology, its structure. Most important part is the electrical activities of the smooth muscle. Depending on this electrical activity of the smooth muscles, the GI tract shows movement, motility, contractility. Different types of movements are there. So these are actually reflection of electrical activity of the smooth muscles. The electrical activity of the smooth muscles are different from nerve cells and the skeletal muscles. We get action potentials in muscles. There are cardiac muscle, there are cardiac muscles, striated muscles or skeletal muscle, and smooth muscles. Where you will get the smooth muscles? Where these are distributed? Most predominantly, you will find them in GI tract. Okay? GI tract has a muscular coat, and in the muscular coat, you learn there are longitudinal layers. There are circular layers of muscles, smooth muscles, and in stomach there are oblique muscles also. Other than this, respiratory tract. You will get smooth muscles in the respiratory tract. You will also get smooth muscles in ureter. The tubular structure coming from kidney to urinary bladder. So there you will also get smooth muscles. You will get smooth muscles in the urinary bladder. You will get smooth muscle along the vascular system, that is arteries and veins. In the wall of the arteries and veins, there are muscles. Beside these, there are smooth muscles in the reproductive organs also, in uterus, in fallopian tubes, in eye. Can you tell me which are the smooth muscles present in eyes? Some muscles contract maintains the diameter or changes the diameter of the lens, thickness of the lens. What are these muscles? Ciliary muscles. Similarly, there are some muscles that dilate, constrict the pupil. How? There is iris. So within the iris, there are some smooth muscles. So there also you will get smooth muscles. You will get smooth muscles in our skin. When we become afraid suddenly, what happens to the hair? They are erected because of contraction of some smooth muscles. These are called piloerector muscles. So we have seen there are a lot of the muscles, smooth muscles are distributed throughout the body. Now these muscles, smooth muscles, are classified into two groups. One is somebody has written unitary smooth muscle. Another multi-unit smooth muscles we'll discuss. Now let us see what is there in a smooth muscle. Then we'll divide. The smooth muscles, their shape is spindle shape. Their shape is spindle shape. The shape is spindle shape. They are non-striated. They are involuntary. You cannot, no, sorry, too early for tomorrow, come tomorrow. Day after, oh, Monday, Tuesday. So, voluntarily, by your wish, you cannot control these muscles. Long fusiform in bundles or fasciculi. What does it mean? The smooth muscles remain in bundle. Some smooth muscles remain in bundle. Remains in bundle. In a bundle, you may get around 1000 cells, particularly in GIT. How many? Approximately 1000 cells in a bundle. Suppose there are 1000 cells in a bundle. So this is one bundle. In our GIT, bundles of such bundles are arranged longitudinally, also circularly. Longitudinally. So initially they are bundled, taking approximately 1000 cells. Now there are so many such bundles, and they will form a sheath. They will form a sheath, and this sheath may run longitudinally. They may run circularly. Let us first see what you will get within these individual cells. You will get plasma membrane, we call it sarcolemma. You will get sarcoplasm. So here the cytoplasm is called sarcoplasm. And at the center you will get nucleus. Beside you will get there mitochondria. You will get there sarcoplasmic reticulum, means endoplasmic reticulum. But they are not so well organized. Here it is poorly organized sarcoplasmic reticulum. What is the function of sarcoplasmic reticulum? What can be the function? What is the function of endoplasmic reticulum? Protein. They are, if it is not smooth, rough, then they will be producing protein. And if it is smooth, they will be produce hormones, steroid hormones like hormones. So their function is different. So they need not produce so many proteins, need not produce any steroidal substances. So that's why it is not so well organized. Moreover, in the muscle cells, particularly in skeletal muscle, we find sarcotubular system. Sarcotubular system? No idea. Okay, when we discuss about the structure of skeletal muscle, then we'll discuss. Then we'll describe. Actually, muscles are having one property, unique property. What is this? Contractility. Only muscles can contract. No other cells can contract. And why they are having this property? Because they are having some contractile protein. What are these? Actin and myosin. Okay. They cannot contract by their own. Somebody has to influence them for contraction. Now, who will influence them? They will be influenced by calcium ion. Now, within the sarcoplasm, cytoplasm, there is no calcium ion. From where it will get this calcium ion? For contraction? Not only calcium ion, it will also need ATP. It will get because mitochondria is there. So ATP is available, energy is available, but calcium ion is not available. Then how to contract? So there should be some provision of providing calcium ion. In our skeletal muscle, there is, for this purpose, there is sarcotubular system. There is T-tubules. Muscle fiber. So these are actins, are jet bands. Here actins are there. And in between myosin is there. From the sarcolemma, some tubular structure enters within the muscle. Enters within the muscle. Tubular structures. They produce branching. And the branch of these, each branch at the end produces a bulbous structure, cistern. Now these cisterns contain calcium ion. Now the second question, these cisterns are not open to sarcoplasm. Now the question comes, how this will be available to the sarcoplasm? That's why we need action potential. We know action potential travels along the cell membrane, along the sarcolemma. So when it is passing through sarcolemma, on the way it will find sarcotubular system. So it will pass along the sarcotubular system and will come into the cistern. When this action potential arrives in the cistern, the calcium ion is released into the cytoplasm. So now this calcium ion will cause contraction, excitation, contraction, coupling. So now the calcium ion will bind with myosin head. Before it should be phosphorylated. So when it is phosphorylated, now it will form a cross bridge. So in presence of calcium ion and ATP, they will form cross bridge. Then these will be formed. Then these cross bridges are formed. Now these threads, they will contract. Now what will happen to the actins? They will be coming closer. What will be happen to the jet bands? These are the jet bands. They will be shortened. So this way, muscle contraction will be made. This is in short, very short. But in between there is some other steps, processes we'll discuss. When calcium ion is displaced from here, then what will happen? These cross bridges will be relaxing, so it will come to its original place. So we can see, for muscle contraction, one thing is needed. What is this? Action potential. So this action potential is important for skeletal muscle, for cardiac muscle, as well as for smooth muscles. Okay, we'll discuss this electrical property of the smooth muscles. Every individual muscle cell, smooth muscle cells. The third point, long fusiform in bundles. These bundles is a fasciculus having near about 1000 cells. When such numbers of fasciculi will be arranged, then they will make a sheet, as I told you. The cells are of varying dimensions. See, in gut, what is their length? 200 to 500 micrometer, and their diameter is 2 to 10 micrometer. Whereas in blood vessels, their length is 15 to 20 micrometer and diameter 2 to 3 micrometer, and uterus 300 micrometer and diameter is about 10 micrometer. The cells contain few mitochondria and centrally placed nucleus. They have poorly developed sarcoplasmic reticulum, I said, and sarcotubular system is present but not well developed as in skeletal muscles. What we'll find here, you will not get these T tubules are there, but in the smooth muscle cells you will not get T tubules. There are small pouch-like invaginations of sarcoma into sarcoplasm called cabuli. This cabuli in skeletal muscle, cisterns is there. In skeletal muscles, in sarcotubular cistern is there. They contain calcium. Here for smooth muscle cells, there is no such cistern. Instead of cistern, what is there? Cabuli. When this cabuli will be stimulated, they will release calcium ion for smooth muscle contraction. Okay? See, these are the contractile elements of a smooth muscle. What are these? Instead of jet bands, here we saw jet bands are there, but here we won't find any jet bands. Instead, you will get what? You will get these dense bodies. These dense bodies are bound to the cell membrane. These are bound to the cell membranes. Here you will find the dense bodies. They will act as jet bands. From the dense bodies, we can see filaments, radiating filaments of actins, thin filament actins, are extended towards a thick filament, that is the myosin. So at the center you can see the myosin. You can see the projections from the myosin heads. The actin will bind with these projections to form cross bridges. So when they will be, these actins will be contracted. The desmosomes, dense bodies will be coming closer, the muscle will be shortened. These dense bodies remain attached to the actin with another tiny filament that is called alpha actin. In smooth muscle, actin and myosin filaments are relatively longer. Relatively means in comparison to skeletal muscle. We said muscles, smooth muscles are two types. What are these? Unitary. Unitary smooth muscle or single unit. Single unit smooth muscle. Now, from your idea, tell me what I have drawn there, whether it is single unit or multi-unit. Multi-unit. Single unit. What is your idea? Somebody said multi-unit. Yes, it can be multi-unit because these cells are separate from one another. For their contraction, since they are not having any communication with other cells, so their contraction must need a nerve fiber. So individual fibers must have a nerve fiber. Then only the individual fiber will be able to contract. It happens in multi-unit smooth muscles. Every muscle cell, smooth muscle, will have their own nerve fiber. But this bundle are not separated from each other. They are connected to each other. Every cell is connected to each other by these junctions. Through these junctions, the electrolytes, ions that are present in one cell that can diffuse to nearby cells. Sodium ions from here to there it can travel. Potassium ion from there to here they can travel through these junctions. Not only this, parasympathetic, they can influence their activity. They can increase their activity, they can decrease their activity, but they can generate their own action potential. Now let's see how they can generate their own action potential, their electrical activity. Autonomic neuron. Every muscle is having nerve supply from autonomic nervous system. Every bundle. Autonomic nervous system means sympathetic and parasympathetic. So this type of neurons will be there. See, these neurons are not penetrating the cells. What they are doing? They are forming a structure. These neurons forming a structure like synaptic knob. We have studied action terminal and knob-like structures. Here, the nerve also produces some knob-like structure. These knob-like structures are called varicosities, swelling. Within this swelling, there is neurotransmitters. Within this swelling, varicosities, there is neurotransmitters. It may be acetylcholine, it may be adrenaline, it may be noradrenaline. Okay? Suppose it is parasympathetic nerve. So what will be there here? Acetylcholine. So when the stimulation is coming into this, so what will happen? From the varicosities, these chemicals will be released into ECF. From ECF, it will enter into the sarcoplasm and it will initiate, it will change the what? Resting membrane. These cells are also having resting membrane potential. Now this neurotransmitter will change the resting membrane potential of the smooth muscles. Okay? So we are coming to now resting membrane potential. Okay? We discussed in our resting membrane potential class how it is developed. Every living cell has resting membrane potential. Okay? Resting membrane potential means you had one question in your exam and 4 marks was there allotted. Who tried to answer this? Somebody got 1, half, not more than this. Those who have answered this. Similarly, if electrical activity of smooth muscle comes in question, as question, you will be answering and answer will be like that only. You are listening, you are not able to hold it, you are unable to reproduce it. This is the quality you are having. This is the sarcolemma of the smooth muscle. Like other cells, inside will be negatively charged. Inside is negatively charged. And outside is positively charged. In nerve cells, so since there is difference in charge across the membrane, so if you place electrode one inside and one to outside and connect it to a galvanometer, what you will observe? A potential change. And this potential change is called resting membrane potential. This potential difference is called resting membrane potential. Okay. For now, what is this resting membrane potential? Minus 70 millivolt. For muscle, it is minus skeletal muscle, it is minus 80 to minus 90 millivolt. But in case of smooth muscle, this potential and even under resting condition is not fixed. It is undulating, varying rhythmic fashion, varying in a rhythmic fashion. So only in case of smooth muscle you will find resting membrane potential not fixed but undulating, rhythmic change in potential. Let us Draw a graph. Time. Millivolt. In nerve, if the nerve is under resting condition, there we find under this resting condition I am recording resting membrane potential. You will find, so after 1 millisecond resting potential is this, after 2 millisecond it is this, after 3 millisecond, after 4 millisecond, after 5 millisecond, after 6 millisecond. So it is not fluctuating. So under resting condition it will remain same. Similarly, in nerve, in muscle, skeletal muscle, you will find resting potential at this level. It is fixed, not changing under resting condition. But in smooth muscle, it is not like that. This resting membrane potential in smooth muscle will fluctuate between these. Means it will fluctuate. Resting membrane potential will fluctuate like this. As time goes, it will show this fluctuating resting membrane potential. Why these fluctuations? Cell is under resting condition. Then why this fluctuation? The fluctuation is because the cell membranes, they are having sodium channels. Every cell is having sodium channels. Sodium concentration is high outside, inside it is less. So sodium ion will always try to enter. If sodium ion enters within the cell, negativity will be increased or decreased? Less negative or more negative? It will be less negative. Means it will be depolarized. Entry of sodium ion means it is being positive ion. Inside is negative, so some negative ion charge will be neutralized. So inside will be less negative. So as a result, what will be there? This is more negative or this one is more negative? Minus 50 or minus 60? Which one is more negative? Minus 60 is more negative. Minus 50 is less negative. Wrong idea you had. So, resting membrane potential will fluctuate like this. This fluctuating resting membrane potential is called slow waves. So what are slow waves? Where you will find? It only in smooth muscle, that too, not in all type of smooth muscle, you will be finding it only in unitary smooth muscles. These fluctuating slow waves only can be seen in unitary smooth muscles. Why these smooth muscles change this fluctuating memory? Because the sodium ion, there are several theories, not yet completely understood. Even now, if you go through a golden book of latest edition, 2024th edition, you will find there also they are telling it is not well understood. However, there are several hypotheses. Somebody says, here the problem is with the sodium channels. Sometimes it opens, sometimes it closes. Opens, closes, opens, closes. So when it opens, sodium ion enters. So it becomes less negative. Again, when it enters, sodium potassium pump activates itself. So again sodium ion is pulled out from the inside. So again it will be coming down. So this is one theory. Another theory says, so there are some sodium calcium channels whose replay opens and closes after a certain period of time. Another theory says it is the function of interstitial cells of Kajal. Mark it, interstitial cells of Kajal. Kazal. We have, it is not there, bundle of smooth muscles. What? Bundle of smooth muscles. We have seen, in between these bundles, there are some special cells. In between the bundles, what?
Interstitial cells.
Cells of Kajal. Kajal. These are also called pacemaker cells. Question become, what are pacemaker cells of smooth muscles? What is their function? Our GI tract. Though we remain in resting condition, it is contracting, relaxing, contracting in a systematic, rhythmic manner. So definitely some stimulation is coming there within these cells. They are causing this rhythmic change in the muscle cells and cause rhythmic contraction. Who are the responsible? these cells. Pacemaker cells you will get in another organ. Where? Your heart is beating only because of this. We have in our body a generator. What is the function of generator? Production of electricity. Though our generator is also, SNOD is also producing electricity. मान लो we don't have power supply from electricity department. So college is having generator. From that generator we are having connections to this light, to this fan. When this generator goes out, there will not be any light, fan will not be moving. So if SNOD fails, our heart will also fail. Since heart muscle will not get electrical impulse for their contraction. But it is not so easy. The God has given you some more provision. So what do we do? Some of you are having 2 aprons. Isn't it? If one is spoiled, then we will use another one. So that you can come into the class wearing the apron. The others who are having one, they will not be able to come. So they will die. But those who are having 2, they will survive. Those who are having 3, they are having more chances. Means, like SNOD, there are other nodes which are also capable of producing electricity. But in presence of SNOD, the other nodes will not act. In absence of SNOD, then only AV node will be functioning. So our heart will not stop. But the efficacy of AV node will not be like that of SNOD. So SNODal cells are also called, this SNOD also called pacemaker. There in the pacemaker you will find some other cells. They are called pacemakers. So these cells generate their own action potential. We will see how these are generated. These action potential are generated. Okay. So in 2 places you will get these pacemaker cells. In intestine or in smooth muscle, these generator cells are called interstitial cells of Kajal. So they lie in between layers of the smooth muscles. So what is there? They have a particular property. What particular property? This rhythmic property, rhythmic change in electrical change across the membrane, resting membrane potential. Sometimes now see, Lakshman Rekha. If this depolarization comes to just below this level, this rhythmicity will be maintained. But whenever you will be touching this Lakshman Rekha, what will happen? Let's see what happens. The interstitial cells of Kajal, they transmitted some impulse to the muscle cells. As a result, These cells will be depolarized, means when stimulation from interstitial cells of Kajal will come to the cells, some calcium channels will open. Before that, it was sodium channels. Now, voltage gated, calcium channels are opened. As a result, lot of calcium ion along with small amount of sodium will be entering within the cell. And as a result, rapid change in potential will be there. So whenever this slow wave is touching this minus 40 millivolt, this is the threshold limit. This Lakshman Rekha is threshold limit. When this resting membrane potential will be touching this, immediately what will happen? A spike will be produced. Because of rapid influx of calcium ion. Voltage will be changed. Voltage across the membrane will be changed. It will become less and less negative, and it will proceeding towards zero, but it will not reach zero level. It will remain within the zero level. So this spike, whenever this spike is produced, before that, with this potential which it is generating, this potential will not be transmitted through the sarcomere to other cells. But when this spike potential is produced, this is like action potential. This is nothing but action potential of smooth muscle. So immediately this will be passing along the sarcomere of the smooth muscle, and it will cause release of calcium, more calcium ion, release of more calcium entering, and the calcium ion enter within the muscle cell, and actin and myosin will contract, will form cross bridge, and there will be contraction of the smooth muscles. So for this, one thing is important, spike potential has to be generated. With spike potential only, within the muscle, the more calcium ion will be entering, and with this calcium ion will cause initiation of smooth muscle contraction. Okay. Now, how many contractions? This will create, since one spike is produced, it will be able to produce one contraction in the smooth muscle. Now see, in case of this, interstitial cells of Kajal influence these slow waves. How it influenced? It made it more negative. More than this. It has crossed just 40. Here it has crossed minus 30, for example. Now what will happen? It will produce spike. More than one, maybe two, maybe three. What will happen? With these three spikes, initially with one spike, one contraction was there. With 3 or 4 spikes, what will happen? There will be more contractions. There will be spasm in the GIT. As it happens when we will face spasm of the GIT. stomach ache, abdominal ache means there is spasm. Rapid in diarrhea. So here contraction, relaxation or movement is faster. That means in the smooth muscles, lot of spikes are producing. Now see the next case. Again I am increasing this. Now it will produce lot of spikes. As a result, what will happen? Now all these spikes will cause sustained contraction. It happens in uterus also. Uterine contraction, as it happens in delivery of the babies, delivery of the fetus. Sustained contraction will be there. So we can see with increase in spikes, there is increase in muscle contraction. In case of these, they are unable to produce any contraction because these are not propagatory in action. So slow waves are not propagatory in nature. Now see, these slow waves, their frequency, in 1 second, how many waves will be there? It depends on area of the GI tract. In stomach, it is 2 to 3. In duodenum, movement is more. In duodenum, frequency is more. It may be 11 to 12. In this frequency, in the terminal part of ileum, it is 8 to 9. So this frequency of waves, slow waves, it varies depending on the zone of the GI tract. Moreover, these waves are influenced by several factors. What are these? Stretch, acetylcholine, parasympathetics. Parasympathetic nerve endings will secrete some neurotransmitters. These neurotransmitters are called parasympathetics. Now this one of the example is acetylcholine. So when these nerves we have seen with bulbous structures, when they secrete acetylcholine, what will happen? This acetylcholine will enter into the cell and will cause, here it was, this wave was between 40 and 50. Now it will be between 45 to 35. Means the level of the voltage, baseline voltage will be going up. It will be less negative. But if the same cells are stimulated by this, norepinephrine or sympathetics, now what will happen? The baseline voltage will be coming down. Means here the slow waves will be there. These cells, under this condition, when this baseline is coming here, it means the smooth muscle cells are less excitable. It is now difficult to make spike potential. When any drug or any agent capable of raising this voltage level to here, the smooth muscle cells in this condition will be more excitable. They can easily form spike potential and can form contraction, can initiate contraction of the smooth muscles. But during tonic contraction, it is prolonged. It can sustain contraction for few minutes to an hour. Okay. Tonic contraction is continuous and can last for several minutes to hours. It is not associated with BER. BER means basic electrical rhythm. These slow waves are called pacemaker potential, slow wave rhythm. All are synonymous. BER, basic electrical rhythm of smooth muscles. It is not associated with BER of slow waves. It could be caused by, number one, continuous repeated spike potential. When the repeated spike, continuous spike is formed, then every spike is producing contraction. So there is no time of relaxation. So there will be sustained contraction. Continuous entry of calcium ion. So from this, what is the name of this? Varicosities contain acetylcholine. What is this? The name of this varicosities? These varicosities. They will liberate acetylcholine and it will cause liberation of, there is one sac-like structure here. From this sac, acetylcholine, calcium ion will be released. Continuous release of calcium ion will be there. As I said, like cistern. In case of circuitubular system, like cistern, there is some inflated portion. Here, calcium ion remains deposited. So this will be, calcium ion will be released from here continuously. It can, because of this, there can be sustained contraction. Okay, so these are tonic contractions. It can be an example of this tonic contraction, uterine contraction during delivery. In our next class, we will discuss what is the mechanism of smooth muscle contraction. So what we learned today? Structure of smooth muscle and mechanism of development of electrical potential. This electrical potential are of two types. One is slow rhythm or pacemaker potential, or BER, basic electrical rhythm. This rhythm, this BER, pacemaker potential, they are unable to produce contraction of the muscle fiber, muscle cells. But when these fibers are capable of producing spikes, then only muscle contraction will be there, otherwise not. These spike potentials are propagatory in nature. This will propagate from one cell to another cell through gap junctions, through sarcomere. Okay, so because of this, all cells will be contracting together. So this is the basic features in short of smooth muscles.